Amino acids are referred to as hydrophobic, which is water-faring, and hydrophilic, which is water-loving. Hydrophobic means that it cannot be dissolved in water, in which it has nonpolar substituents, which basically help it do that. And hydrophilic means that it has polar R groups that form hydrogen bonds to water. Amino acids also have both a carboxylic group and an amine group. These functional groups can act as buffer sites. At a certain pH, the amine group gains a positive protonated and negative charge, which is deprotonated. Overall, if an acid is added, the amine can pick up extra protons, and if a base is added, it can neutralize, which is similar to the behavior as a buffer. That's how aqueous solutions of amino acids are buffer solutions.
Okay, so now we're going to be discussing optic activity in terms of, in relation to amino acids. So basically, objects that have non-superimposable images are chiral. And so what do I mean by that? So basically, if you take like any object and you reflect it across, say, a mirror, and you can impose that image directly onto the object, then it's superimposable. But if you can't, then it's not. So basically, this ideology can be applied to molecules. And so if a molecule's mirror image is different than the molecule in 3D space, then the molecule is chiral. So shown here is an example of a chiral molecule. So basically, you can see this isomer 1 is reflected, and the reflection is isomer 2. And so because isomer 2 cannot be superimposed upon isomer 1, the molecule is chiral. So optical activity is the ability of a chiral molecule to rotate the plane of plane polarized light. And so basically, what we really want to determine is whether or not an amino acid is optically active. So if an amino acid is chiral, then it's optically active. And so actually every single amino acid except for glycine is optically active. And so glycine is not optically active because it's not chiral. So as you can see here, it has 3 different types of chemical groups that are attached to the center carbon. And so it's always going to end up being superimposable. So because of that, it's not going to ever be chiral, and so it's not going to be optically active. So here are some examples of some optically active molecules. And so these amino acids are optically active because they're chiral. So as you can see here, if you just kind of, imagine a mirror, basically, you can see that, like, you wouldn't be able to superimpose that image upon the amino acids, so they are optical. So peptide bonding is basically, a peptide bond is basically just a bond between 2 amino acids, and polypeptides form chains of amino acids with peptide bonds. So an example of this is the reaction between glycine, serine, and alanine to form a tripeptide. So as you can see here, these attach, some of the double bonds become single bonds as the amino acids create peptide bonds with one another and form a polypeptide.
So what are proteins? A protein is a natural polymer that performs many functions in our bodies. The building blocks of proteins are monomers known as amino acids. These monomers are called amino acids because they belong to the amino group NH2. And in amino acids, an NH2 molecule is always going to be attached to a carbon atom. There are 3 main different types of protein structures. The first is a primary structure. The primary structure is the order or sequence of the amino acids in a protein chain. This primary structure is indicated by the 3-letter codes for the amino acids. For example, the primary structure of a tripeptide containing lysine, alanine, and leucine would look like this, which is represented by the shorthand notation lys ala lu. Now, this shorthand notation is referencing specifically the order they are. If the alanine was before the lysine, it would be ala lys lu. Or if the leucine was before both of them, it would be lu lys ala. Whatever order the amino acids are in, that is what the shorthand notation is. The number of possible structures in an n-number peptide is n factorial. So if you have 3, there's going to be 6 possible structures. If there's 4, there's going to be 24 possible structures. The second type of structure is secondary structure. The arrangement of the chain in a long molecule is the secondary structure, and it is determined by the hydrogen bonding between the lone pairs of an oxygen atom in the carbonyl group of an amino acid and the hydrogen atom attached to a nitrogen of another amino acid. As seen by the diagram, you can see how each one of them is bonded around the molecule. This diagram is what the secondary structure is about. The last structure we want to talk about is the tertiary structure, and it's the overall shape of a protein. These come in a variety of different shapes, such as long, narrow, or globular, maintained by interactions such as hydrogen bonding, dipole-dipole interactions, ionic bonds, covalent bonds, and London dispersion forces. As you can see in the figure, all these tertiary structures have their names. A is ionic, B is shown through hydrogen bonding, C is what a covalent bond does, D is London dispersion forces, and E is a dipole-dipole bond.
How does denaturation affect the function of a protein? Well, denaturation causes a protein to lose its biological function. And even though the second and tertiary structures might be altered, the peptide bonds, which belong to the primary structure, are left intact. And furthermore, denaturation does not hydrolyze the peptide bonds too of a protein. Carbohydrates are biologically important molecules. They serve as a food source for most organisms, and it has a formula of CH12. The monomers of monosaccharides are also called simple sugars, and moreover, by combining these monosaccharides, such as sucrose, a disaccharide is formed. Disaccharide sucrose is composed of glucose and fructose by elimination of water to form a carbon to oxygen to carbon bond. These bonds between the rings are glycoside linkage, which was explained earlier.
So on to monosaccharides and polysaccharides. So basically, glycoside linkage is the bond formed between multiple monosaccharides. Alright, and starch, which is a polysaccharide, which is made up of multiple monosaccharides, has low solubility in water. And it's because it contains amylopectin, which is a highly branched polymer of alpha glucose, and amylose, which is a straight-change polymer of alpha glucose. And so the bond between the chains of monosaccharides forming starch require too much energy for the solvent-solute interactions to overcome. So because of that, starch has actually pretty low solubility in water unless you heat it. Yeah. So there's some general differences between starch, cellulose, and glycogen. Starch is a polymer of alpha-D-glucose. It consists of amylose and amylopectin, and it's used in plants for cellular fuel. Cellulose, on the other hand, is a polymer of beta-D-glucose, and it's a major structural component of woody plants and natural fibers. Glycogen is a polymer with a similar structure to amylopectin, but it has more branching going on. And it's also the main carbohydrate reservoir in animals. And so basically, the branching in the glucose allows the rapid breakdown of glycogen, which can be converted into glucose, which is used by mammals for energy. So here's the structure of amylose and glucose, and you can kind of imagine the structure of glycogen. It will be like the same as amylose, but it's gonna have some more branching going on. So yeah, A is the structure for amylose and B is the structure for glucose in the diagram below.
Let's talk about the main structural differences between DNA and RNA. With DNA, which is deoxyribonucleic acid, DNA has a double helix structure that has 2 complementary bases on the 2 strands. It is also a very large molecule. It is massive. And RNA is a singular strand of the DNA structure, and it's significantly smaller in some cases. Nucleic acids are DNA and RNA, and the monomers of these nucleic acids are things called nucleotides. Nucleotides are composed of 3 parts. The first part is a 5-carbon sugar, deoxyribose in DNA, and ribose in RNA. The second part is a nitrogen-containing base. This is cytosine, adenine, guanine, thymine, or uracil. And last, there is a phosphoric acid molecule, H3PO4. The compounds cytosine, adenine, guanine, and thymine are known as nucleic acid bases. And these nucleic acid bases form hydrogen bonds with their structural counterparts. This hydrogen bond is what makes them bases.
Bases of DNA are formed with hydrogen bonds that are bonded to one another. Cytosine is bonded to guanine, and adenine is bonded to thymine. However, the hydrogen bonds are found as pairs on the 2 strands of DNA. During cell division, the cell unwinds and the new complementary strands are constructed. These unraveled strands serve as a template for attaching complementary bases, which result in the double-helix structure of DNA.
So during cell division, the DNA unwinds completely. And so basically, new complementary strands are constructed on the unraveled strands, and so all these strands contain the DNA bases. So basically, these individual bases on the strands, they serve as a template for attaching complementary base pairs. And so the pairs are going to be connected based on the previous rules. So like cytosine with guanine, adenine with thymine, etc. And so here's like a kind of a diagram that shows the process whereby one strand within the DNA is unraveled, and then subsequent complementary pairs are attached, and basically this will form an entire new strand of DNA. So you're going to start with 1 and end up with 2 basically.
Protein synthesis is a major function of DNA. Protein synthesis occurs when a section of DNA called a gene contains the code for a specific protein. These codes transmit the primary structure of the protein, the sequence of amino acids, to the construction machinery part of the cell. There's specific code for each amino acid, so it assures that the correct amino acid is inserted as the protein chain grows. A code consists of 3 bases, is called a codon. And just know that a DNA holds the genetic information while the RNA transmits the actual information.
A segment of DNA is also called a gene. A gene contains a specific code of 3 bases called a codon. With a base substitution mutation, which is also called deletion mutation, only a single nucleotide is changed. Therefore, only 1 codon is affected. Since only 1 codon is affected, it does not constitute to such a serious mutation or even fatal. However, if a base were to be deleted, which is also called a frameshift mutation, the DNA template strand gains more of a wrinkle and results in a nucleotide to be omitted from the replicated strand. This results in a complete alteration, and this is what constitutes to a fatal mutation.